Bio 139 lectures, blood vessels. In this video, we are going to talk about the type of blood vessels. Blood vessels, they form a closed delivery system that begins and ends at the heart, and they carry the blood from the heart to the cells of the body, and they carry the blood back from the cells of the body to the heart, and they also work with lymphatic system to circulate the fluids. We have arteries. Arteries are those blood vessels which carry blood away from the heart. And all the arteries, they contain oxygenated blood other than the pulmonary arteries and the umbilical vessels of the fetus. They are different. And then we have capillaries. Capillaries, they are in direct contact with the tissue cells. This is the place where the exchange of the gases and the nutrients happens. And then we have veins. Veins are the vessels which carry blood back towards the heart, and all the veins of the body, they contain deoxygenated blood except the pulmonary veins and the umbilical vessels of the fetus. Arteries, when they leave the heart, as they move along, they become smaller and smaller. We have elastic arteries. Aorta is an example of an elastic artery. They are also known as conducting arteries. These arteries have relatively big lumen, and from the name you can tell that it has more elastic fibers. And as it moves down, then muscular arteries are smaller than the elastic arteries, and they have more muscular component in them, but they also have elastic fibers in them. Similarly, as they move along, they become smaller and smaller till they become arterioles. Arterioles, they are very small arteries, and they are known as the resistance vessels. Why? We will talk about it. And then these arterioles, they make the capillary bed. They divide into capillary bed. This is the arterial end of the capillary bed, and this is the venous end of the capillary bed. And then these capillaries, they merge to form postcapillary venule, and then those merge to form bigger veins, and they carry blood back to the heart. And then along with these blood vessels, we also have lymphatic vessels, because we know that around 3 liters of blood every day, it is it just it is present in the interstitial fluid. It is unable to dissolve back into the venous end of the blood. So this is carried by the lymphatics, and these kidney bean shaped structures on the pathway of the lymphatics, they are known as lymph nodes. So lymphatics, they carry this blood which is in the interstitial fluid back to the circulation, and it puts it in on the venous end of the circulation. Now let's go over the structure of the blood vessel wall. So we know that all the vessels, they have a lumen, because they are going to carry the blood back to the heart. So all the vessels, they have a lumen. That lumen is surrounded by a wall. So these walls are made up of 3 layers. The innermost layer is known as the tunica intima. Then there is tunica media. Then there is tunica externa. So the exception are the capillaries. Capillaries only have endothelium, which is lying on a basal lamina, because capillaries, they are very thin. They are where the exchange is going to happen. So we do not want a thick wall in the capillaries. And remember, endothelium, it is the epithelium which lines the vessels. The innermost layer of the blood vessel wall is the tunica intima, and you can remember it that this is in contact with the blood. So this is more intimate, most intimate to the blood. So this lumen is lined by the tunica intima. Tunica intima consists of endothelium. Endothelium is the epithelium which lines the vessels. And what is the type of that endothelium? It is simple squamous epithelium, and it is continuous with the endocardium, which is lining the inside of the heart, and it is very smooth from inside, because we do not want any friction in the blood vessel. Otherwise, if there is turbulence in the blood, there can be problem. And then under the endothelium, so this is the endothelium. Under the endothelium, there is a layer which is known as subendothelial layer. Subendothelial layer, it has connective tissue basement membrane. So endothelium, then we have subendothelial layer. and it is found only in vessels larger than 1 millimeter. So all these layers, tunica, intima, media, and adventitia, according to the type and size of the blood vessel, they will be different from each other. They will have some difference. Inside tunica intima, we have tunica media. Tunica media is composed of smooth muscles and it contains elastin fibers. And it has sympathetic vasomotor nerve fibers. Because this layer, it can constrict and it can dilate. So when these muscles contract, there is vasoconstriction. Vasoconstriction, it decreases the lumen diameter of the vessel. So this diameter will become smaller. The lumen will become smaller when there is vasoconstriction, and then they can also cause vasodilation. Vasodilation means the lumen, it will become bigger. And this is the bulkiest layer, and this is responsible for maintaining the blood flow and the blood pressure. Outside tunica media, we have tunica externa. You can remember it from external. So it is the outermost layer of the wall. It is also called tunica adventitia. It is composed of connective tissue. There are collagen fibers that protect and reinforce it to the surrounding structures. And this layer, it is infiltrated with nerve fibers and lymphatics. And then the larger veins, they also contain elastic fibers. Like I already said, in different types of blood vessels, these layers will differ. Some component will be more in some will be less in some. In some, this tunica media will be bigger. In some, other layers will be more bigger. So there are some differences according to the type of the vessel, but this is like the general plan. Then there are vasa vasorum. What are vasa vasorum? This is the blood supply of the vessel. So vasa vasorum basically means vessels of the vessel. These are tiny vessels which supply the outermost external layer of the blood vessel. And where does the internal layer, like the intima, it gets its nutrition from? From the blood which is in the lumen. Let's go over this diagram. This is how the artery and the vein, they appear under the microscope. Just notice that the artery, it has a uniform lumen. It is kind of more oval or more rounded, whereas the vein, the lumen is kind of collapsed. So this is how the arteries and the vein, we see them in the microscope. And this is because the veins, they have thinner wall compared to the arteries. Now let's go over the three layers and let's see the differences between the artery and the vein. So first of all, we have tunica intima. Tunica intima, it consists, it can have endothelium. It will always have endothelium. Actually, all the vessels have endothelium. Then a subendothelial layer, then internal elastic membrane. Now let me go on the arterial side. So this is an elastic artery. If this is an elastic artery, it will have all the three layers. Okay, so it has an endothelium, then it has subendothelial layer, then it has internal elastic membrane. So now let's go to the vein. So this vein has endothelium, then it has subendothelial layer, and it has no internal elastic membrane, right? So there are differences in the vessels. There are differences between arteries and veins, and then there are also differences between different types of arteries and different types of veins. So now let's go to the tunica media. So tunica media, it consists of smooth muscles and elastic fibers. So this is the layer with smooth muscles and elastic fibers. And outside this layer, there is external elastic membrane in the artery. So now let's look at the vein. In the vein, we have this smooth muscle layer, and there is no layer of external elastic membrane, right? Now let's go to the tunica externa. Tunica externa has collagen fibers and vasa vasorum. So both of these features are present in the arteries and in the veins. And now there they have made up, before I move on to the next slide, look at this capillary. Capillary is only endothelial cells which are resting on a basement membrane. So these are the capillaries. And then notice there are valves in the vein, because veins, they have to take blood back to the heart. Okay, now let's look at the differences in different types of the vessels. Let's look over arteries first. So this is elastic artery. In elastic artery, the endothelium is present and it is like the normal size. So endothelium, and then there will be subendothelium, and then the elastic tissue. It has a large amount of elastic tissue, then smooth muscle, and it also has nice amount of smooth muscle, and then the fibrous or the collagenous tissue, this is also present in this much amount in the elastic arteries. Now let's move to the muscular artery. The muscular artery also has endothelium, although it is a little lesser than the elastic artery. Then it has elastic fibers. They are less compared to the elastic arteries, but it has those. And then look at the smooth muscles. Its smooth muscle layer, it will be in tunica media. So its smooth muscle layer is very much more bigger compared to the elastic artery. And then also in the adventitia, it has more collagen tissue. Now let's move to the arterioles. Arterioles, they are smaller than the muscular arteries and they are known as the resistant vessels. So see, the endothelium is almost the same as the muscular artery. The elastic fibers, they have decreased further, and the muscle layer, it is also smaller but it is present. And then this is the collagen fiber. Now let's go over to the venous side. So they have started at the capillaries. Capillaries, we know we only have endothelium, right, and the basement membrane. There is no elastic tissue, no smooth muscle, no collagen fibers. Then these capillaries at the venous end, they make venules. Venules are the smallest type of veins. So they have endothelium, and now they have some smooth muscles, and then they have some collagen fibers. And then the bigger veins, they have endothelium. Now they have some amount of elastic fiber in the vein, in the bigger veins. Then the smooth muscles are much more compared to the venules, and then the collagen fibers, they have more collagen fibers than the venules. So I put this slide just to show you the difference. This is how arteries appear under microscope, and this is the collapsed vein. So now let's go over different type of arteries. We know that arteries, they are divided into three groups based on their size and functions. First of all, we have elastic arteries, then muscular arteries, and then arteriole. Now, the first type of arteries are the elastic arteries. They are thick-walled arteries and they are near the heart. Aorta is example of elastic artery, aorta and its branches. And they are low resistance pathways. And they are also called conducting arteries because they conduct the blood from the heart to the muscular arteries or to the medium-sized arteries. And elastin is found in all the three layers, in tunica media, tunica intima, tunica adventitia. I did not say them in order. And they have got a nice amount of smooth muscles, but they do not vasoconstrict. This is important. And they act as pressure reservoirs. What does that mean? When the heart beats and pushes the blood in the vessel, they expand, and then between the heartbeats they contract to maintain the blood flow, because now the blood flow has stopped. Just imagine if you put a hose to a tap. When you open the tap, the blood is flowing, but just imagine the tap is like a heart. It pushes the blood in the hose and then it pauses, then it pushes again and then it pauses. So because with every beat the heart is pushing blood, so between the pause, when it will pause, there will be no flow in that hose. But elastic arteries, because they're elastic, between the beats they come back, they recoil, just like elastic, when we stretch it recoils. So that maintains the speed of the blood flow. Our second types of arteries are the muscular arteries. Elastic arteries, they give rise to muscular arteries. They're also called distributing arteries because they deliver blood to body organs, and they have different diameter range, and they account for most of the named arteries. For example, if I say like brachial artery, it is a muscular artery. And they have the thickest tunica media because the smooth muscle layer, it is present in the tunica media. So it is very normal for us to think that muscular arteries have the thickest tunica media with smooth muscles. And they do have elastic tissue, but it is less compared to the elastic arteries. And then they are active in vasoconstriction. They vasoconstrict when they get the The signals, when they are stimulated, they vasoconstrict. It means they make their their lumen narrow. Vasoconstriction is when the vessel, it contracts and the lumen becomes narrow. And then these muscular arteries, they become smaller and smaller and smaller till they become arterioles. Larger arterioles, they contain all the three tunics: tunica intima, tunica media, tunica externa. Smaller arterioles are mostly a single layer of smooth muscle surrounding endothelial cells. So in the smaller arterioles, there will be endothelium, then there will be smooth muscle layer, then there will be, that is it. So this is how smaller arterioles are, and they control the flow of blood in the capillary bed. They can vasodilate. Vasodilate means they can make their lumen larger, or they can vasoconstrict. They can make their lumen smaller due to the smooth muscle present in them. And this arteriolar diameter, it varies in response to changing neural, hormonal, and local chemical influences. So they are influenced by the neural stimuli, by the hormonal stimuli, and by the local chemical stimuli. They are also called resistance arteries because changing diameters changes the resistance to blood flow. So just imagine water. If it is going through a narrow pipe, there is more resistance. But if that pipe, the lumen becomes, the diameter becomes bigger, then the resistance will decrease, and then they lead to capillary beds. Capillaries, they are very small microscopic vessels. Their diameters are so small that only a single RBC can pass through. So just imagine RBCs are making a line and passing through the capillaries. And capillary walls, they have a very thin tunica intima only. There is endothelial cells which are resting on a basement membrane. And one cell forms the entire circumference. They are so small. And then there are some cells known as pericytes. What are pericytes? They're spider-shaped cells. They're present on the outer surface of the capillaries, and they are part of the contractile stem cells that can generate new vessels or scar tissue. So when there is an injury, these are the stem cells, and they can generate new vessels and the scar tissue, and they help stabilize the capillary wall since they are so thin, and they also control the permeability of the capillaries. And I already said that they play a role in vessel repair. Okay, and capillaries, they supply almost every cell of the body except for cartilage, epithelia, cornea, and lens of the eye. They have some other mechanisms. You can go over them if you're really curious. And then the function of the capillaries is to exchange materials like gases, nutrients, hormones, and all the stuff which needs to be exchanged. It happens at the level of the capillaries between the blood and the interstitial fluid. We have three types of capillaries. There is one thing common in all these three types of capillaries. They have tight junctions with gaps. Those gaps are known as intercellular cleft. So let me show you. So look at this first type, the continuous capillary. So these are endothelial cells which are attached to each other through tight junctions, and then this is the intercellular cleft. So these endothelial cells, they are not joined all over. There are clefts in between. And now over here, see, these are endothelial cells. These are endothelial cells, and they're joined by tight junctions. Over here as well, these are endothelial cells, endothelial cell, endothelial cell. They are joined by tight junctions. Okay, now let's go over. So our first type is continuous capillary, then we have fenestrated capillary, then we have sinusoid capillary. So let's see. So as we are moving down, they are becoming more like permeable, more wider. So let's see the continuous capillary. Continuous capillaries are the least permeable and most common. Okay, they are abundant in skin, muscles, lungs, and CNS. Often have associated pericytes. So this is the pericyte, the cell I mentioned that is a stem cell, and it is a repair cell. It repairs and it contracts and it helps. Okay, so this is the pericyte on the outer surface of the capillary. And then they are the most continuous capillaries. They have intercellular clefts. These are intercellular clefts between the endothelial cells. Okay, but these clefts, they are absent in the brain. So this is where the blood brain barrier is formed. So these clefts are tight, they are closed. So the endothelial cells are tightly closed in the brain, and this makes the blood brain barrier. So this was about the continuous capillaries, the most common, the least permeable, and they are associated with pericytes, and they have clefts in the brain. The clefts are absent. There are tight junction all around the cell, and that makes the blood-brain barrier. Now let's go to the fenestrated capillaries. Fenestrated capillaries have large fenestrations or pores that increase the permeability. So see, these are the fenestrations or pores in the endothelial cells, and these are because of these they have increased permeability compared to the continuous capillaries, and they occur in areas of active filtration or absorption, or in the endocrine hormone secretion where it is required. So where does filtration happen? Filtration happens in the kidneys. So these fenestrated capillaries are present in the capillaries, and in the intestines we need to absorb the nutrients, so they are present in the small intestines. And then where the endocrine hormone secretion has to take place, because those hormones, they have to go in the blood. And these fenestrations are Swiss cheese like holes that funnel through the endothelial cells. So this looks like Swiss cheese. Fenestrations are usually covered by a very thin layer of condensed extracellular glycoprotein. This layer has little effect on solute and fluid movement. Okay, so there is a membrane covering it, but it does not hinder the solute moving from across the endothelial cell or the fluid. And in some digestive tract organs, the number of fenestration and capillaries, it increases during the active absorption of nutrients. Okay. All right, then we come to the third type, the sinusoid capillaries. Sinusoid capillaries are the most permeable, and they occur in limited locations. Okay, so where are they present? They're present in liver, in bone marrow, in spleen, and adrenal medulla. So these are the only places where sinusoid capillaries are present. They have large intercellular clefts as well as fenestrations and few tight junctions. So this, see, if we can even see here, they're so large. So this is like cut it, if I cut it at this level. So this is how it will appear. There is a large intercellular cleft between two endothelial cells, and they have fewer tight junctions. Tight junctions are present, but they are fewer. And they have incomplete basement membrane. Okay, they have incomplete basement membrane because they are very permeable, and they are irregularly shaped and have larger lumens than the other capillaries. Blood flows slowly through the tortuous channels. Macrophages may extend processes through the clefts to catch or prey the foreign organisms. So macrophages are present around them. Now let's see what are capillary beds. Capillary beds, they are interwoven network of capillaries between arterioles and the venules. And then there is a term microcirculation. Microcirculation, it is the flow of blood through capillary bed from the arteriole to the venule. So this blood going from here to here, this is known as microcirculation. And then we have a type of vessel known as terminal arteriole. It is branch of the arteriole that further branches into 10 to 20 capillaries. So this is terminal arteriole, and it is dividing into a number of capillaries. And capillaries, they are the exchange vessels. And exchange of gases, nutrients, and wastes from the surrounding tissues, it takes place in the capillaries. Then these capillaries, they drain into post-capillary venule. So this is post-capillary venule, and this is terminal arteriole. And this in between is the capillary bed. Blood flow through the capillary bed, it is controlled by the arteriole. When the arteriole is dilated, it means that its lumen, the diameter of its lumen is bigger. More blood comes in and more blood goes into the capillary bed. And when the arteriole is constricted, the blood it is kind of shunted away from the from going into the capillary bed. So this arteriole it can monitor the blood flow in the capillary bed, and it is under the influence of vasomotor nerve fibers and local chemical conditions. So local chemical conditions and the vasomotor nerve fibers, they can either make the arteriole to dilate or they can make the arteriole to constrict, and as a result the blood flow in the capillary bed, it can easily flow through it or it can even stop. If it really constricts, there will be no blood flow going to the venule. So the regulation is at the arteriolar end, not at the venule end. So our body uses this mechanism of increasing the blood flow through a capillary bed or decreasing the blood flow through a capillary bed. Let me give you a common example from every day. So when we exercise, more blood goes to our muscles and less blood goes to our digestive system because I'm working hard and my muscles need more blood. So what happens that the arterioles which are present in the muscles, they dilate and more blood is sent there, and whereas the arterioles which are present in the digestive system, they constrict, so less blood is going to the intestine. And vice versa, when I eat a big meal, the arterioles which are present in my intestines, they increase the blood flow in the capillary bed, whereas at other organs, for example my muscle, they don't receive that much blood at that time. There is a special type of capillary bed present in the mesenteries. What are the mesenteries? They are the serous membranes that hold the intestines. And what is that? So just look at this. This is an arteriole and this is a terminal arteriole. So blood is coming in through the terminal arteriole, but instead of going in the capillary bed, it is being shunted. It is being shunted straight to the venule. And this vascular shunt, it is made of meta-arteriole. So this is known as meta-arteriole, this one. And then this channel is known as thoroughfare channel. So instead of going to the capillary bed, it is going straight to the venule through this pathway, meta-arteriole and thoroughfare channel. This is known as vascular shunt. And why is it, how is it possible? There are precapillary sphincters. These precapillary sphincters, they are closing these pathways. So the blood, it is going straight there. And these precapillary sphincters, they are not innervated and they are controlled by the local chemical conditions, but they are only present in the mesenteries of the intestine. We are done with the arteries. Now let's go over veins. Veins carry blood towards the heart. Okay, and the formation begins when capillary beds, they unite in post-capillary venules and merge into larger and larger veins. Venules, there are smallest venules, which are the post-capillary venules. They are the smallest form of venules. And the capillaries, they unite to form the post-capillary venules. These consist of endothelium and a few pericytes, and they are very porous. They allow fluid and WBCs into the tissues. In inflammation, if you remember, there is margination, which is adhesion of WBCs to the post-capillary venule endothelium, which is followed by diapedesis. What is diapedesis? Their migration through the wall into the inflamed tissue. So these structures, they are very porous. And then the larger venules have a thin tunica media. Now the muscle is coming in as the veins are becoming larger. One or two layers of smooth muscles are present in the larger venules, and they have a thin tunica externa as well. So after from the capillaries, the capillaries merge and they form venules, and then the venules, they merge and they form bigger and bigger veins. And the veins, they have all the tunics: tunica intima, tunica media, and tunica externa. But their walls are thinner and they have larger lumens compared to the arteries. in histological preparations, veins are usually collapsed and their lumens appear slit-like, as you can see in this slide. Tunica media is thin, but tunica externa is thick in the vein. So tunica media is thin, but externa it is thick. And the tunica externa, it contains collagen fibers and elastic fibers. Large lumen and thin walls, they make veins good storage vessels. What does this mean? It means that when there is large amount of blood present in the vein, they can expand and they can store the blood. So they are called capacitance vessels or blood reservoirs, and they contain up to 65% of the blood supply of the body. This is just a slide for comparison. I will not go over it again. We discussed it. And this, where you can see the different membranes of the vein. If you want to uh, revise, I will not go over it again since I already went over it. And this is the different types of veins. And now over here you can see that almost 60% of the blood, it is in the vein, it is in the venous system in our body. And then 15% is in the arteries and arterioles, 12% is in the pulmonary system, and then 8% is in the heart, and 5% is in the capillaries. So this is where my blood in my body is right now. This is the proportion. A few more points about veins. Blood pressure is lower in the veins compared to the arteries. So veins, they have to make sure that the blood, it reaches back to the heart. So how do they compensate for that lower blood pressure? They have larger diameter lumens, so the resistance in the veins is less. And then other adaptations are there. These veins have valves. These valves, they prevent backflow of blood. Valves are most abundant in the veins of the limbs, especially in the legs, because there the blood has to go up against the gravity. So these valves, they prevent the backflow of the blood. And these valves, they're absent in the thoracic and abdominal cavities. Then we have another feature about the veins. There are venous sinuses. What are venous sinuses? They are flattened veins with extremely thin walls. Their walls only have endothelium, and these veins, because they have very thin walls, they are supported by the tissue around them. One very common example is the venous sinuses in the skull, in the brain. We have venous sinuses. These are very dilated veins with very thin endothelial lining, and they're supported by the tough dura mater that covers the surface of the brain. Now, what can go wrong with the veins? So varicose veins, it is a condition in which there are dilated, painful veins due to incompetent valves. Factors that contribute, they include genetics and the conditions that hinder the venous return. For example, if someone is standing for a very long time, there is pooling of blood in the veins. And if that person also happens to be obese, there will be problem in that blood returning to the towards the heart, going up, because the fat, it compresses on the vessels. Then in pregnancy, this can happen. And then elevated venous pressure can also cause varicose veins. For example, during delivery, when the woman has to strain a lot, then because of the increased venous pressure, it can also cause varicose veins. So there is a term anastomosis. What does this mean? Our vessels, they anastomose. Anastomosis, they are interconnections of the blood vessels. So blood vessels, they connect with one another. There can be an anastomosis between two arteries. For example, this is one artery. It can communicate with another artery. So arteries, they can communicate with each other. Similarly, there can be connection between an artery and a vein. It is known as arteriovenous anastomosis. Then there can be an anastomosis between two or more veins. So anastomosis, they are interconnections between the blood vessels. Let's quickly see what is arterial anastomosis and what is the advantage. So at arterial anastomosis, it provides alternate pathway to ensure continuous flow. So in simple words, many organs, they are supplied by more than one artery, and these arteries, they interconnect with one another. Now if one of the artery it gets blocked, the blood supply to that part of that organ can come through the other artery. So these anastomosis, they're commonly found in joints, abdominal organs, and brain and heart, but they're absent in retina, kidney, and spleen. So let's talk about the bad news first. For example, one of the artery of the kidney, it becomes blocked. Now that portion of the kidney is going to die because there is no collateral or anastomotic channel coming to that part of the kidney. So now in brain we have this. This is the blood supply of the brain. We are not going to learn the names of the arteries just so that you can understand. So this is coming from the left side from the carotid artery. This is from the right carotid artery, and then we have basilar arteries. All these arteries, they're forming a ring. Also the arteries which are coming interiorly, they all form a ring. So now this part of the brain is being supplied by this artery, right? So the blood which is coming from here, it goes here. So just imagine if this gets blocked, the blood to that part of the brain can be supplied from this artery. From this artery, it can go there. So this is the advantage of this collateral channel. Now these anastomosis, they can also happen between an artery and a vein. The example of the arteriovenous anastomosis we already went over. In the mesentery, we have this shunt where a metarteriole, it is directly taking blood to the thoroughfare channel without, and it is skipping these capillaries. So this is example of arteriovenous anastomosis. Then the anastomosis, they are more common in the veins compared to the arteries. So this is diagram of a hand, and this is how the veins, they anastomose with one another. So this is the end of our anatomy of the blood vessel.